Good morning. Welcome to Murmu Biology. Have you ever thought of before reaching the antibiotic or the medicine that you eat, whether it be an antibiotic or it be an analgesic, as simple as or any paracetamol that you eat, is before incoming it into the exploring it into the market, it undergoes animal trials, isn't it? So an more effective and non harming way of doing animal trial is trying it on cells, isn't it? So let's see question number 69, which is related to tissue culture, isn't it? So question says, which of the following method is most appropriate for long-term preservation of animal cell lines with minimal genetic drift? Minimal genetic drift means mutation kind of thing, or deviation of inheritance material, that is DNA, the pattern of DNA, the genome, to something else, isn't it? So the options are continuous subculturing, cryopreservation in liquid nitrogen, serum starvation, and feeder layer culture. So let's see what this question says. So continuous subculturing, if you know a little bit about microbiology, then you know that the cell can be cultured in a culture media of agar-agarose, isn't it, having nutrient and other media in the, it can be cultured in the culture plate or it can be cultured in the broth, liquid broth, isn't it? So continuous subculturing means the continuous subculture of from the parent cell to the next generation for research purposes, isn't it? That is cell passing from one generation to next generation. It gives the subculturing gives a gives an option or gives an opportunity for cell from going one generation to next generation, isn't it? Second option is cryopreservation. Cryopreservation is the method of preserving the cell, isn't it? It is stored in liquid nitrogen, that the temperature of liquid nitrogen is minus 196 degrees Celsius. So the cell is being stored and kept in the sub-zero temperature, isn't it? Third option is serum start. Serum start when we grow cell in the culture media, isn't it? So the animal cell is provided with serum, isn't it? Serum is a component of blood, isn't it? You know, when the cell is allowed to sit, allowed to clot, the liquid which remains, the watery type liquid which comes upon the after clotting the red blood cell, the liquid which is floats above the media is known as serum, isn't it? So when we and that is a very blunt type of definition. High rich media serum is available in the market, which is being used in the culture, isn't it? So serum start is the culture cell is deprived of the serum, and due to this deprivation the cell undergoes senescence, isn't it? That is, cell gets stuck at a particular cell phase, isn't it? May it be G0 or may it be G1 or G2, so that the scientist could undergo or detect the cellular functions at that particular stage of the cellular cell cycle phase, isn't it? And the fourth option is feeder layer culture. In this option, the stem cell, as you can see, feeds upon a feeder layer cell, isn't it? So a bed is prepared of the cell which will act as food or which will act as nutrient to the current cell. You can see in the option here, feeder layer herbs is prepared which are purple in color, and human cell has been kept upon the feeder layer cells, so that act as the purple cell act nutrients for the human ES cell, isn't it, which are and growing on the feeder layer cell, isn't it? It is a kind of maintenance kind of thing, isn't it? So you have seen these four what do you say options. You have seen continuous subcultural, that is growing next generation of the cell. You have seen serum starvation, in which the culture is starved or the cell is starved at a particular cell stage. You have seen feeder layer culture. In this also the cell is continuously developed, but in only in cryopreservation the cell is stopped and all the metabolic activities are being uh, temporarily stopped, and the cell is preserved for a long term storage purposes, isn't it? So you can see this in long term stability. Cryopreservation is used for this halts the cell division, isn't it? And in continuous subculturing, the active expansion is going on, because it is the cell is dividing and producing next generation cell, isn't it? And serum starvation is temporary state of synchronizing cell cycle. All the present cell in the serum in the culture stay at a particular cell cycle stage to for the respective study, isn't it? And co-culture state is, that is feeder layer culture is used for used as a food for the particular cell to grow upon, isn't it? So these are the application. And tissue culture, integrated cell culture workflow. You can see the cell is thawed. Cell is thawed means the cell is defreezed from the cryopreservation. It is acted upon the primary culture, and then it undergoes quality control, and it has a feeder layer culture, and it has experimental manipulation, and the serum is stored for particular study, and the experimental assay is done to read what the study is saying. Again, the particular cell is scaled up and preserved in the cryopreservation. So this is a typical workflow of the cell culture. Here you can see the application of that this cell culture workflow can be used as a in drug efficiency toxicity setting. Instead of using the whole animal, the cells are being tested. In it can be, this workflow can be used in gene editing, CRISPR, isn't it? It can be used for signal pathway. This experimental design can be used for cell differentiation induction or disease modeling kind of thing. Okay, so it is again the literature part. So seeing upon the question, which of the following method is most appropriate for long-term preservation of animal cell lines with minimum genetic drift? So here you have seen that continuous subculture is making the cell divide more and more, isn't it? It is from parent cell, the cell is continuously dividing. In serum starvation also, the particular cell is stored, but for a short term time, isn't it? And feeder layer culture is also, the cell is growing, isn't it? And only in cryopreservation long term storage is present, and the cells are stopped at a particular stage, at a particular all the metabolic activity are stopped. So in all three options, that is subculturing, serum starvation, feeder layer culture, there is a chance of DNA replication taking place, isn't it? Because the metabolic activities are continuously going on. But in cryopreservation in liquid nitrogen, the cells has been stopped, and the metabolic reactions, that is DNA replication, transcription, this translation, these have been stopped. So there is no question of minimal genetic drift, that is deviation of the genome that the cell contains, isn't it? So this is again a diagram to look upon And these are the option: continuous subculture, serum starvation, feeder layer culture, and cryopreservation. Okay, this is the literature. You can see cryopreservation is the gold standard for long-term storage, and these are why other options are not suitable for this question. And you can donate rupee 1, okay, to support us and help us root grow with us. If you like the video, then like, subscribe, share, or download the for offline viewing. And thank you. Stay tuned to Murmu Biology.